Hello everyone and welcome to our channel, MooTechie. It's wonderful to have you here as we dive into the fascinating world of molecular analysis. Today, we're going to explore the powerful techniques that allow scientists to see the hidden structure of molecules. Get ready for an exciting journey through light, mass, and magnetic fields. Now, let's move on to our overview. Overview of UV visible mass and NMR spectroscopy. This presentation covers the principles, data interpretation, and practical applications of these methods. Next, we explore our introduction to these fundamental techniques. Spectroscopy studies how light or energy interacts with matter to reveal molecular structure and composition. These methods are non-destructive, highly sensitive, and provide complementary data for a complete analysis. While their mechanisms differ, all three techniques produce a spectrum where peaks serve as the primary readout for identification. And in modern chemical analysis, we typically combine data from all three methods to solve complex structural puzzles. Let's briefly meet our three main characters. UV visible spectroscopy measures electronic transitions, often related to color and conjugation. Mass spectroscopy determines molecular mass and fragmentation patterns. And NMR spectroscopy maps the carbon-hydrogen framework and its connectivity. Coming up, we'll discuss the first technique in more detail. UV visible spectroscopy, principle and theory. Molecules absorb specific photons in the UV-vis region, causing electrons to jump from ground to excited states. Quantification follows the Beer-Lambert law. That's A equals epsilon BC. Absorbance A is directly proportional to concentration C. Functional groups called chromophores, like a carbon-carbon double bond or a carbon-oxygen double bond, determine where these absorption bands appear. Spectra are characterized by the wavelength of maximum absorption, lambda max, which is unique to a compound's electronic structure. Now, let's visualize a typical spectrum. This image shows a UV visible absorption spectrum with wavelength in nanometers on the x-axis. We see two prominent peaks, one at a lambda max of 450 nanometers and another at 200 nanometers. This is a typical UV-vis absorption spectrum where the prominent peak indicates the lambda max, which is used for both identification and quantitation. Following this, let's understand what's happening during these absorptions. Absorption and electronic transitions. The introduction here mentions intriguing a dye and lists a series of energy levels from N1 to N22, representing the quantized energy states electrons can occupy. Here are the key concepts. UV-vis spectroscopy excites valence electrons from occupied orbitals like sigma, pi, or n orbitals to unoccupied sigma star or pi star orbitals. Common transitions include the high-energy sigma to pi star transition and the lower-energy n to pi star or pi to pi star transitions seen in conjugated systems. When a molecule has extended conjugation, it reduces the energy gap, shifting the lambda max to longer wavelengths. This is known as a bathochromic shift. Finally, band intensity and peaks are governed by selection rules and a property called molar absorptivity, epsilon. There's also a diagram described as a molecular orbital energy diagram. It illustrates the relative energy gaps between different orbitals and shows the corresponding electronic transitions that happen when a photon is absorbed. As we continue, let's see how this theory is applied in the real world. UV-vis practical applications. Quantification is achieved using the Beer-Lambert law by measuring absorbance at lambda max to build standard calibration curves. In kinetic studies, we monitor the change in absorbance over time to determine reaction rates and mechanisms. The pharmaceutical industry relies heavily on this for quality assurance and quality control, using precise peak analysis to verify drug purity and concentration compliance. And in colorimetry and metal ligand complex studies, scientists utilize shifts in lambda max to analyze binding interactions. let's look at a common graph used for quantification. This chart shows a standard calibration curve. It plots absorbance on the y-axis against concentration in micromolar on the x-axis. We see a series of experimental data points and a straight best fit line running through them, with an R-squared value of 0.999, indicating a very strong correlation. This is how we convert an absorbance reading into a concentration value. Now let's shift our focus to a different but equally powerful technique: mass spectrometry basics and principle. Mass spectrometry works by ionizing chemical species and sorting the resulting ions based on their mass-to-charge ratio, or M to Z. The process occurs in three main stages: the ionization source, the mass analyzer, and the detector. The resulting mass spectrum is a plot of ion abundance versus M to Z ratio. And the distinct peaks you see represent the molecular ion and characteristic fragments which are used for identification. Let's break down those three stages. First, ionization. Here, sample molecules are converted into charged gas phase ions, for example, via electron impact. Second, mass analyzer. Here, electric or magnetic fields separate ions according to their M to Z values. And third, detection. The ions strike a detector, creating a signal that is proportional to their abundance. And the final output is a mass spectrum, which is a graph of intensity versus M to Z, showing those distinctive peaks. Now let's see how this separation actually works: the M to Z concept and ion separation. This slide has a diagram illustrating the process. It starts with sample ionization, followed by ion acceleration, then magnetic field separation, and finally detection. Ions with different M to Z values like 50, 75, and 100 are separated and detected. The resulting mass spectrum is shown as a graph with peaks at these M to Z values, labeled relative abundance. Now let's talk about the core parameter. The mass-to-charge ratio M to Z represents the mass M divided by the charge Z. Since most ions are singly charged, meaning Z equals 1, the M to Z value typically equals the molecular mass itself. Isotope patterns are a key clue. Elements like chlorine or bromine create distinct M+2 peaks due to their natural isotopes, which really aids in identification. Precise calibration is crucial. It ensures that the measured M to Z values align with true atomic masses for accurate formula assignment. And the principle of separation is that ions are deflected by magnetic fields based on their momentum and M to Z value before they hit the detector. Next, we explore how to read the story a mass spectrum tells us. Mass spectrum interpretation. We are presented with a sample mass spectrum. It's a plot with relative intensity in percent on the y-axis and M to Z on the x-axis, ranging from 0 to 120. Key peaks are visible at M to Z 43, 71, 75, 85, and a prominent one at 100, which is labeled as the M+ peak, representing the molecular ion. So how do we analyze this? First, locate the molecular ion peak M+. This is the peak at the highest M to Z, which in this case is 100, and it typically represents the compound's molecular weight. Second, identify the base peak. This is the tallest peak in the spectrum, set at 100% relative abundance, and it represents the most stable fragment ion. Third, analyze the fragmentation. Specific neutral losses like M-15, which suggests loss of a methyl group, can reveal structural connectivity. And fourth, check for isotopes. Look for small clusters of peaks near the M+ peak, like M+1 or M+2, as these can indicate the presence of elements like chlorine or bromine. The figure caption reminds us that this is a mass spectrum displaying relative ion intensity versus mass-to-charge ratio, and that our key task is to identify the M+ parent ion and rationalize the fragment peaks. Now let us look at our final core technique, which provides incredible structural detail. NMR Spectroscopy Introduction and Principle. Nuclear magnetic resonance relies on the properties of nuclear spin, for example in hydrogen-1 or carbon-13 nuclei, which align in a strong magnetic field. The local electronic environment around each nucleus shields it differently, causing nuclei to resonate at unique frequencies when they are irradiated. This frequency difference relative to a standard called TMS is termed the chemical shift, delta, and it's measured in parts per million. The key outputs from an NMR spectrum are the peaks, which tell us about identity and position, integration, which tells us the count of nuclei, and splitting patterns, which reveal connectivity. There's a simple diagram illustrating the basic NMR principle. It shows a magnetic field, B, and nuclei aligning and resonating at specific frequencies after a radio frequency pulse is applied. Coming up, we'll understand what the chemical shift scale is telling us. Chemical shift representation. Figure 1 shows a simulated proton NMR spectrum. From right to left, we see the TMS reference peak at 0.0 ppm, then a cluster for alkyl protons. Further downfield, a set of aromatic protons, and finally a peak way downfield for an aldehyde proton. So let's understand this scale. The chemical shift scale, delta, is measured in ppm relative to the TMS standard reference at 0.0 ppm. Electron withdrawing groups de-shield protons, shifting their peaks downfield to higher ppm values, as we clearly see with aldehydes between 9 and 10 ppm. Conversely, electron-rich environments like those around alkyl groups are shielded and appear upfield between 0 and 2 ppm. These distinct functional group regions allow for rapid structural mapping based purely on peak position. The discussion note confirms that this figure illustrates the correlation between the electronic environment and the chemical shift, from shielded alkyls to de-shielded aldehydes. To finish our look at NMR, let's decode the conversation between protons. NMR peak analysis and splitting patterns. The N+1 rule is a fundamental concept. It states that a signal is split into N+1 peaks, where N is the number of equivalent protons on adjacent carbons. The coupling constant J, measured in Hertz, defines the distance between the lines in a split signal and indicates the bond connectivity between protons. Integration calculates the area under a peak, which is directly proportional to the number of equivalent hydrogen atoms producing that signal. And common splitting patterns you will encounter include singlets for no neighboring protons, doublets for 2 neighbors, triplets for 3 neighbors, and quartets for 4 neighbors. This slide includes a visual guide to NMR splitting patterns. It shows examples of a singlet, a doublet, a triplet, and a quartet. A specific example is given. A proton with 2 neighbors, following the N+1 rule, would result in a triplet, represented as 3 peaks. The text confirms that these are typical splitting patterns observed in proton NMR spectra, perfectly illustrating the N+1 rule. Now let's step back and compare all 3 techniques side by side. Technique comparison. This is a comparative analysis of their measurement principles, outputs, and utilities. First, UV visible spectroscopy. It measures electronic transitions of conjugated systems upon light absorption. Its data output consists of broad absorption bands defined by their absorbance and position at lambda max. Its primary strength is highly accurate quantitative analysis via Beer's law and conjugation detection. It requires UV transparent solvents and the presence of chromophores. Next, mass spectrometry. It measures the mass-to-charge ratio of ionized molecules and their fragments. Its data output is a spectrum of intensity peaks plotted against M to Z. Its primary strength is exact molecular weight determination and revealing structural fragmentation patterns. It requires an ionization source and high vacuum conditions. And finally, NMR spectroscopy. It measures the magnetic resonance frequencies of nuclei like hydrogen-1 or carbon-13. Its data output consists of signals defined by their chemical shift, delta, their integration, and their splitting. Its primary strength is providing the most detailed structural insight, including connectivity and even stereochemistry. It requires deuterated solvents and a strong and stable magnetic field. To bring everything together, let's review our key takeaways. Summary and key takeaways. This has all been about a unified approach to molecular structure determination. UV visible spectroscopy primarily measures absorbance at a specific lambda max. It's essential for quantifying concentration and identifying conjugated electron systems. NMR spectroscopy maps the carbon-hydrogen framework. It uses chemical shift,
to determine the electronic environment and signal splitting to figure out neighbor connectivity. And mass spectrometry separates ions by their M/Z ratio. It provides the precise molecular mass and reveals structural clues through characteristic fragmentation patterns. And the most important message is on integrated analysis. No single technique is sufficient for complex unknowns. Combining electronic, mass, and magnetic data provides a robust, cross-verified structural solution. Thank you. See you in the next class.